You've seen a little bit of the Myelin architecture. Matt will show you how to measure myelin with MRI scan, as well as use multimodal imaging to parcellate the brain.
Okay, yes, so I will be talking about non-invasive cortical parcellation with Myelin maps and other MRI modalities, talking about methods, results, and tools. The topics for today are the non-invasive MRI-based methods for mapping cortical myelin content, then the HCP's multimodal cortical parcellation, from which we've used based on group average gradients. And then we'll talk about individual identification of cortical areas in individuals based on those multimodal aerial fingerprints and parcellation validation. And since the course is about tools, I'm going to try after each topic to tell you about the tools that already exist and about those that are in development and what data you would want to acquire to be ready to use them. So first, non-invasive myelin mapping. We've heard a little bit about this already from Simon, but the first guys who were doing the histological myelin mapping of the cerebral cortex were Oscar and Cecile Vogt, and they were among the first brain parcellators in the early 1900s. And they recognized distinct cortical areas based on several myeloarchitectonic parameters, including the overall myelin content, which basically would be the average of this myelin-stained piece of a section here, the number of and strength of the tangential bands, which are indicated by the red, the bands of Baillargeon, the density of radial fibers that point from the white matter up to the peel surface. And the Votes thought that each cortical hemisphere contains around 200 myeloarchitectonically distinct cortical areas. Based on what we know from comparing monkeys to humans so far, somewhere around 150 to 200 cortical areas is about right. So how do we do this? What are the contrast mechanisms we use in MRI? So myelin has several properties that make it visible to MRI. So here we have a myelinated axon and an EM. The myelin wrappings here are the dark stain. Myelin is rich in lipids. It's co-localized with iron, particularly within the cortical gray matter. And it restricts the motion of some nearby water molecules. And so these properties lead to several forms of MR contrast, T1 contrast, T2 star contrast, and magnetization transfer contrast, respectively. And each of these contrast mechanisms has been verified by comparing myelin-stained sections to MRI-based myelin maps. And these are some of the studies that have actually done this, where they've scanned the same tissue, they scanned some tissue and then they stained it. What about the approach that we use in the Human Connectome project for myelin mapping? Well, we use the ratio of T1 weighted MP range divided by T2 weighted space. And this actually makes use of all three forms of myelin contrast. So just to illustrate how this works, here we have a volume slice of a T1 weighted image. This sulcus, big sulcus here, is the central sulcus. We saw quite a bit of the central sulcus in Simon's talk. So we have white matter is bright white. The white matter surface is the green outline. CSF is on the outside of the peel surface, the blue outline. So our gray matter ribbon is running in between these two surfaces here. And if you look within this ribbon, just in the T1 weighted MRI image, you can see that the intensity is brighter here, say on the anterior bank of the central sulcus, than it is here in frontal cortex or down there in parietal cortex. If we look in a T2 weighted image, though, we get the opposite pattern. So now the things that were brighter in the T1 are now darker in the T2 weighted image. And it's these differences in intensity that we're going to use to measure cortical myelin content. So because the T1 and T2 images have inverse contrast, we can divide the T1 by the T2, which enhances our contrast and removes the receive field and some of the transmit field. And now we can see, we can colorize it, and we can already see cortical areas just in the volume slice here. So we've got primary motor cortex is this thick, heavily myelinated area on the anterior bank of the central sulcus. Anterior to that is area 6. Then here in the fundus here is area 3A. Area 3B is thin, but also heavily myelinated here on the post-central bank. And then behind that is area 1. So we can really improve our ability to analyze this across subjects and visualize it if we map the pattern in the volume onto each individual's cortical surface. So basically this white line here is where the volume slice intersects with the surface. And generally we're mapping the average myelin content across the cortical layers. In the HCP, we're at 0.7 millimeter isotropic T1 and T2, and we can see some hints of the actual laminar myeloarchitecture. Maybe you can see something a little bit right down there, for example, but we don't yet have enough resolution to be able to reliably map those type of laminar patterns. So what can we see in an individual subject with a myelin map from the Human Connectome Project? Well, there are a variety of cortical areas, areal features that are visible. We've already seen the primary somatosensory and motor cortex that was visible. Early visual cortex is visible. Early auditory cortex. The MT plus complex is also heavily myelinated in red here. I don't know if it's completely, there it goes. Retrosplenial cortex. Here, basically, right above the splenium of the corpus callosum is a very heavily myelinated cortical area. Here is actually a white matter pathway that goes through the gray matter, the perforant path, and we can see that in the subiculum. The frontal eye fields, the VIP-LIP homologue, what we think is the VIP-LIP homologue here in the IPS. We can also see very lightly myelinated cortical areas, such as the anterior insula, anterior cingulate, and the temporal pole. And in between these regions, we don't have quite so much contrast for myelin. So the next several topics are actually going to be how do we subdivide these oceans of blue here. But first, let's just see how we can use this technique to compare homologous areas across humans, apes, and monkeys. So here we have those 3 species. We've got group averages of 196 humans from the Human Connectome Project, 29 chimpanzees and 19 monkeys from the Yerkes Primate Research Center. And we've scaled these images according to their volume. So human brains are obviously much bigger than chimp brains, which are, again, much bigger than monkey brains. So we can see many of the same heavily myelinated features across subjects, primary somatosensory motor cortex. We see it in all 3 species. Early visual cortex, early auditory cortex, the MT plus complex we can nicely see across the species. Retrosplenial cortex, the perforant path, the frontal eye fields. So in monkeys, this is the LIP VIP complex, and we see something that seems to correspond across species, which is why we think this might be the human LIP VIP complex. Also, we can see these very lightly myelinated regions in black, in dark purple, anterior insula, anterior cingulate, and temporal pole across all 3 species. But what's really different across these species are these oceans of blue here. So in a monkey, we've got a relatively small region here that expands from monkey to chimp, and then dramatically again from chimp to human in the inferior parietal cortex. Similarly, in the lateral temporal cortex and the prefrontal cortex. And these regions that have expanded so dramatically is where a lot of the human-specific specializations, behaviors like language and tool use tend to be centered in these regions that have expanded so dramatically. So how have we validated our approach neuroanatomically? So here we're going to focus again on the central sulcus. So this is a group average myelin map. And if we want to see where are potential cortical areal boundaries, we're going to focus on the locations where the myelin content changes most rapidly. And that's what this gradient tells us. Basically, this is the first derivative, so it's a rate of change map, and the reds and yellows and greens are where the myelin content is changing. We've drawn these white markers along these gradients, and they're going to be the same across the panels. So we can see how they delineate where myelin content is changing. And we've compared this data to the probabilistic cytoarchitecture. The difference here between, it's the same data that Simon was showing us, but the important difference here is this data was mapped onto the cortical surface from each individual subject by Bruce Fischel and aligned across subjects on the surface, which is the same method that was done here for the myelin maps. And so we have area 4, area 3A, 3B, 1, 2 here. And just like the myelin maps, we can see where does the probability change most dramatically. We can take a gradient of the probabilistic maps, and we see that the gradients of the myelin map and the probabilistic map nicely agree. So that means we can feel pretty confident that the myelin map is telling us area 4 here, this stripe of very heavy myelination. Then 3A next to that. In the fundus, more light myelination, 3B on the other side, more heavy myelination, and so on. And we see this in the right hemisphere as well. So take-homes from myelin maps, how to make them. So what brain images should you acquire? Well, you want to get a high-resolution T1 and T2, and high resolution means 1 millimeter isotropic or better. But really, we found that we get much higher quality individual subject maps if we're at 0.8 millimeters or better. In the HCP, we did 0.7. That requires a 32-channel MRI coil, though. It's recommended that you actually get a field map to help distortion correct these two images. The effect isn't huge, but when we're talking about myelin mapping in some areas, it makes a difference. It's not like fMRI, where it's a much bigger effect, though. And we have some example MR protocols posted on the Human Connectome Project website, which you can look up to see basically what contrast parameters do you want to use to do this. To produce the maps, the analysis software, so our structural minimal preprocessing pipelines generate myelin maps using the latest code. So if you just run those, you get a lot of additional things, but the myelin maps will just come out of them. And you can download those. This website is also linked to from Human Connectome Project. But the download and the instructions of these pipelines are here. There are some other approaches to myelin mapping that give very similar maps, but they are using quantitative methods such as T1 maps, T2 star maps, or magnetization transfer maps. And so these are all quantitative measures as opposed to relative measures. And it's definitely fine to use them if they're a very important part of your study. But generally, they require more scanning time or a higher field strength to achieve similar contrast to noise and resolution. The reason we prefer the T1 and T2 approach is because it's a little bit quicker to get us be able to find cortical areas with. All right, so now let's talk about multimodal cortical parcellation with group average gradients. So a few preparatory remarks about this. Basically, how do we get our multimodal MRI data ready to do a precise cortical parcellation? I'm actually going to talk about these topics in more detail this afternoon. I'm trying to keep the lectures have different content in them. So I'll only give the briefest discussion of them, and if you're more interested in these topics, come to this in the afternoon. But they were very important to the precision of what I'm going to show you. So we need to minimize sources of blurring in our data by using careful processing and analysis techniques. And we really need to avoid poor cortical registration, doing smoothing of our data, or other kinds of lossy processing. So for cortical data, that really means we need to analyze and visualize data on surface models instead of 3D in the volume. And in particular, we need to use cortical surface registration across subjects and studies to be able to maintain the ability to see the boundaries in the places they were in the original data. Also, we should drive surface registration using features that are more closely tied to cortical areas, such as Myelin maps or resting state networks, instead of folding patterns, because as Simon said, often the macroanatomical landmarks the folds are relatively poorly correlated with where cortical areal boundaries are. So we want to drive our, Michelle, maybe I need your pointer now. This one's dead. We need to drive the registration with aerial features. And so there's a new surface registration algorithm, MSM, that can do this. Also, it's important if we're comparing across studies to remove any group average registration drift, and I'll talk more about that in the afternoon. So data that has been brought into the HCP's SIFTI gray ordinate standard space and file format form a really good basis for cortical parcellation. What is that? So it's basically how we can do better than a volume-based analysis for the whole brain. So we're going to consider gray matter structures according to the geometric model that is best suited for each, and that means surfaces for the sheet-like cerebral cortex and volumes for globular subcortical nuclei. So here's a schematic of the gray ordinate space. So a gray ordinate basically refers to a coordinate that can be either a surface vertex. Here we have 30,000 surface vertices for the left hemisphere, 30,000 surface vertices for the right hemisphere, or subcortical voxels, and we've got 10 subcortical structures here that come from free surfer. So we end up with a total of 90,000 gray ordinates when we've got our data sampled at a 2 millimeter spacing. We're going to register individuals' cortical data using nonlinear surface registration and their subcortical data using nonlinear volume registration. And if we do our analysis in the gray ordinates framework, we can greatly reduce the analysis-induced uncertainty in spatial localization. So tools to get you into the SIFTI gray ordinates framework. Again, that's one of the major goals of the HCP minimal preprocessing pipelines is to get your raw imaging data into SIFTI in the standard gray ordinate space. The MSM algorithm, which I mentioned very briefly, I'll talk more about this afternoon, is actually going to be released in the next version of FSL, I hear, talking to Mark Jenkinson. And we're going to try and get our aerial feature-based surface registration pipeline, the HCP's pipeline called MSM also released. If you want to visualize SIFTI data and do analysis on it, Connectome Workbench, the HCP's visualization tool, the command line tools, and MATLAB can all analyze SIFTI data, and the next release of FSL will also have some SIFTI capabilities in some of the binaries, and more will be coming in future versions. The data, the next HCP data release is actually going to provide multimodal data aligned using the aerial features in addition to the folding-based aligned data, so you actually be able to analyze data that's already been aligned using aerial features. If you want to, you're going to want to use this approach on your own data. The images that you'll want to be acquiring are a T1 weighted image, a T2 weighted image, a field map for distortion correction, particularly of the fMRI, and then a fair amount of fMRI data. It could be resting state data. It probably can also be either task or even movie data. Importantly, you don't just want 5 minutes of fMRI. You probably need something 30 minutes or more. In the HCP, we're getting an hour. And you want to have high spatial and temporal resolution. And these are actually the same requirements for these pipelines. Basically, the imaging requirements are always going to be the same. It's T1, T2, field map, and fMRI. There are a variety of other software that you can use for surface-based analysis. FreeCipher actually makes our surfaces in the HCP minimal preprocessing pipelines. AFNI Summa, if you're more familiar with AFNI, or even CBS tools are very good for 70 MP2 range surface-based analysis. So on to parcellation now, how one might parcellate the brain. So historically, as we were hearing from Simon, brain areas have generally been defined using invasive methods by transitions in one or more neuroanatomical properties. Simon talked a lot about architecture, but also you can use function, and classically that might have been recording from electrodes that are placed in different locations in a monkey brain. Connectivity, you can make injections and see where do the resulting tracers go and map out areal boundaries with connectivity. And also topographic maps, maps of the, for example, the visual field space or the body surface or sound frequency space. These all fall into topography, and generally we think of cortical areas as having each cortical area, say each visual area, having a map of the visual field. So the HCP is measuring each of these properties non-invasively in 1,200 subjects. And today I'm going to be focused on the cerebral cortex for multimodal parcellation. So most extant parcellations were generated with only a single areal property or modality because that was all that was available or easy to use. But in the HCP, we can actually use multiple modalities at the same time to generate a cortical parcellation. And we're going to, as we saw earlier, we're going to use gradients. Basically, where do these modalities change across the surface as an objective measure to highlight potential areal boundaries? So what makes a gradient convincing as an areal boundary? Well, the most important one that Simon alluded to earlier is if we've got a transition in architecture or function or connectivity that's located in the same place as another modality. So if we've got agreement in the spatial location of the border between two or more independent modalities, that really gives us confidence that we've probably got an areal boundary there. If it's present in both hemispheres, that's less likely to be a chance occurrence for sure. Obviously, if we don't have a known imaging artifact, such as susceptibility, dropout, or something like that. If we've got prior literature evidence for the boundary, that gives us more confidence as well. And the final step in brain parcellation is to relate the spatial relationships of areal boundaries to existing parcellations, trying to figure out what is the name of the area. Is this an area that somebody's described before, or is it a new area? So we've already seen the myelin maps here. Again, we're focused on where does myelin content change across the cortical surface, and the spatial gradient tells us objectively where the transition in myelin content occurs. And the local maximum of the gradient is the most likely location of a potential areal border. Some transitions you can see are larger than others, but it's those transitions that occur in multiple modalities that are especially interesting as areal border candidates. We can also see transitions at cortical thickness. That also gives us architectural information. One really nice example is in the central sulcus between the posterior bank, which is very thin, and the anterior bank, which is much thicker. So we could see sharp transitions in thickness right along that boundary there. Yeah, this advancer, I'm pressing it about 3 or 4 times each time to get one thing. Also, we're regressing the curvature of the surface out of the thickness map because generally gyri are thicker than sulci, and that's not the effect we're interested in here. We're interested in differences in cortical thickness that are related to brain areas. We can use functional imaging maps. So this is a contrast map from story versus rest contrast. Obviously, the positive areas have more activity during the task, and the negative areas have more activity during rest. And just like the architectural measures, we can take a gradient of this map and see where is the task activity changing most rapidly across the surface. Connectivity is very important for finding areal boundaries. So we've got a seed for this functional connectivity map right up here. And the regions that are yellow are functionally connected to this location. The regions that are blue are anti-correlated. And the gradient tells us where the functional connectivity changes across the surface and by how much. So we're starting with a seed location there, but now we're going to step across the gradient. We can see the functional connectivity pattern changes dramatically, and then we step again and it changes again. So I will try to go back, and you can see these changes. And interestingly, if you focus on the areas that are connected in this network, these same areas are coactive in this language task here. I'm not going to talk much about topographic connectivity. I'll go into that in detail in the afternoon lecture. But task fMRI and resting state connectivity can also be used to define areas using topographic maps. And retinotopic maps are a great example of this. And so we made use of those maps in the following parcellation I'm going to show you. Right. So let's put it all together for one cortical area first before we look at the multimodal parcellation. We're going to focus here on this strip of lightly myelinated cortex here in between the frontal eye fields up there and the premotor eye field there that are more heavily myelinated. And so we're going to draw these white dots along the myelin gradients. These are most likely areal boundaries. And then we'll look in our task data, and we can see there's a unique task pattern of activation in this area versus its neighbors. And so not surprisingly, the task fMRI gradients line up with the myelin gradients. What about connectivity? Well, we could put a seed down here in another part of this task activation network, and we've got unique functional connectivity to that location. But if we put a seed in a different area, here, this heavily myelinated spot in the IPS, we get a very different pattern of functional connectivity, basically an absence of connectivity. So not surprisingly, the functional connectivity gradients are very strong in this location, and they line up with the myelin and task gradients. So we've got multiple independent modalities, architecture, function, and connectivity that all agree on an area. And the last step of the parcellation is to try and figure out, has somebody seen this area before? So identify the area with respect to the extant literature. And one really nice candidate for this area is from an older parcellation by Adolf Hopf. They call this area 55B. And so this is actually one of the first, actually the first myelin map where they actually tried to map out myelin density and show it on the surface. And this is based on histology and a painstaking drawing. But we could see this area here has slightly less myelin content than its neighbors on either side, which makes it a good candidate for being the same as what we see in the imaging data. So lots of work to do for 150 to 200 cortical areas in each hemisphere, but it can be done. And one last point before we get to that is topographic subareas in somatosensory and motor cortex. So we can very nicely with architecture define areas in the central sulcus using myelin and thickness, the primary motor and sensory areas. But if we look in functional connectivity, just the full correlation level, the strongest boundaries are actually sort of perpendicular to these areas. And that's because connectivity is stronger between basically the same part of a map than, say, connectivity between the foot and the face. That's weaker than connectivity between face motor and face sensory. And functionally, we could see these somatotopic boundaries. If we measure them with task fMRI, we see nicely the same boundaries as the resting state. So we call these areas sub-areas. So the multimodal cortical parcellation, just to set the stage, We constructed this from 210 HTTP subjects brought into standard gray ordinate space using MSM aerial feature-based registration. We defined aerial borders using gradients and architecture, Myelin maps, and thickness maps with the curvature regressed out, function, task fMRI maps, connectivity, resting state, functional connectivity, and topography. And then we tried to identify the areas with respect to the prior neuroanatomical literature, keeping the same names when possible. So here's our folding map here. We had a few hypotheses going into this. Our qualitative predictions based on monkeys and partial human parcellation from a few years ago was that we'd find somewhere between 150 to 200 cortical areas per hemisphere. There'd be wide variability in aerial size and shape. And there'll be examples of interareal heterogeneity, such as those early somatosensory motor topographies. Do we really not have another slide advancer? I'm having to press it 5 times to get it to respond. Okay. So here's the multimodal cortical parcellation here, where we've got the boundaries shown on the surface. This is our inflated surface in our flat map here. And we were able to see quite a few cool things. We've got our sensory motor strip here. We've got our early visual areas, just as many other people have seen. This one's not doing anything. Oh, there it goes. Yeah, maybe it's a little bit better. So qualitative results, we found 178 areas and complexes, potentially containing multiple areas per hemisphere. We got wide variability in aerial size and shape. And we got some of these topographic sub-areas. So here we've colorized the parcellation based on the 3 main input streams to the brain, visual, auditory, and somatosensory are blue, green, red. And then the 2 most dominant cognitive networks, task negative and task positive, are bright and dark, respectively. And basically, the core areas are pure colors, and then the areas that are sort of in between, either being visual or somatosensory, for example, or auditory and visual, are going to be mixes of colors. What kind of analyses can we do now that we've got a parcellated, nice parcellation? So this is the parcel boundaries overlaid on the Myelin map here. And what I'm going to do is flip over to basically a parcellated Myelin map, which is where we take the average Myelin content within each parcel. And we see that the map doesn't really change all that much in terms of its pattern. So if we go between one of these and the other, you lose a little bit of detail, but not actually that much, which is really striking given that we've gone from basically in this single hemisphere around 30,000 vertices all the way down to 178 areas. We could do other kinds of parcellated analyses, like make parcellated connectomes. This is with full correlation where we seeded V1 back here. We can also do a partial correlation where we basically, each pair of areas here, we find the unique connectivity between each pair, say between V1 and V2, where we've controlled for the connectivity of every other area. So we regress out. We have 178 areas. We regress out the time series of 176 of them, leaving only the pair that we're correlating. We could see a nice connectivity between V1 and other areas in the visual system. Other parcellated analyses, well, this is going to be another example with functional connectivity. So if we put a seed here in the PGI, in the middle of the task negative network, and that's the full correlation. And the partial correlation, we can see nicely this network lighting up very specifically. We can do task fMRI analyses in parcels as well. So here we're going to take our our time series, and we're going to average a time series within the parcel, and then we're going to run our task analysis on that. So we get big benefits in improving our SNR by doing this, because we're being able to average across regions that are reasonably homogeneous in terms of the task fMRI signal. And so there's two contrasts, working memory up here and the language story contrast down here. And these are all of the Human Connectome Project's task contrasts in a matrix. The line here is which contrast you're viewing. One can think of, once you're thinking in terms of parcellated analyses, you can think of, say, the HCP's MRI data, the multimodal MRI data, as a 3D matrix that has parcels by features, by subjects in it. It's a very compact, manageably sized, high SNR data set. And it helps address multiple comparison issues that you have with voxelized data. So you might have 90,000 gray ordinates or 200,000 voxels in M&I space, but we're probably only talking about 500 at most brain areas. We've found 178 in each hemisphere, maybe 100 or 100 subcortical areas as well. So you could apply this concept to your own data. And the concept of parcels by features by subjects we'll look at in the next section as well. So we've done this at the group average level. Now can we identify these cortical areas in individuals, and how do we validate the parcellation? So I've only got time for 3 quick validation points. So it is possible to train a supervised classifier, which is actually Carl Hacker's perceptron that he's going to tell you a lot more about in his lecture at the end of this course, to identify these cortical areas in individuals based on their multimodal fingerprints, the pattern across these multiple modalities. We can also make individual subject parcellations automatically now that we have a group parcellation. And this includes identifying areas that are misaligned despite our best efforts at registration. So we find that some areas and some subjects just simply have different areal topologies from the group. And I'll show you an example of that. We can make probabilistic maps of cortical areas, since we're able to identify them in individuals. And using only the trained classifier and the dense multimodal data, we can reproduce the parcellation in a completely independent HCP data set. So we basically so we used 210 subjects to make the parcellation and train the classifier, and we held out an additional 210 subjects for statistical validation. And all we're going to bring from the 210 we used for parcellation is those aerial fingerprints, no spatial information, aside from a very coarse localization I'll show you. And also that pairs of neighboring cortical areas have statistically significant and large differences across multiple modalities. So let's recall what makes a cortical area distinct. A cortical area should have a distinct pattern in one or more of architecture from its neighbors, of architecture, function, connectivity, and topography. And we can combine all these into a multimodal aerial fingerprint for each cortical area. We can make some reasonable assumptions about where a cortical area is located. For example, we don't ever expect V1 to be located outside of the occipital lobe. And we'll use these neuroanatomical properties to find group-defined cortical areas in individuals. So how do we do this? How do we learn aerial fingerprints to delineate and identify cortical areas in individuals? Again, Carl Hacker will tell you about his classifier in detail, the methods of it. So I'm just going to tell you about how we changed the modifications we made. He worked with me on this from his application of 7 resting state networks to my application. So we're not going to try and classify all 178 areas at once. That's a very hard problem for a classifier if you have that many classes. Instead, we're going to do something simpler. We're going to classify each area from its 30 millimeters surrounding it. So basically 30 millimeters in every direction along the surface we're going to consider. And so this is that really coarse spatial localization that we have, but it's within what's reasonable if you've done a registration, in spite of those misalignments. We're going to combine across classifiers a little bit later. And instead of using functional connectivity maps like Carl is going to tell you about, we're actually going to use multimodal features in the classifier. So we're going to use Myelin thickness, task FMRI, and resting state connectivity and topography. We'll use not quite as many hidden nodes because we're only doing a binary classification. So this means we're going to have one classifier for each area, and then we're going to combine across classifiers. So the first question is, does the classifier identify misaligned cortical areas? So we've got, here we're going to look at a Myelin map. Here's our folding map, and these are two resting state networks. This one is a language-related network. This is from a D40 ICA, and here's more of a dorsal stream visual network here. So if we run the classifier, these are the outputs that we get from it. So these are areal probabilities that vary from 0 to 1 here. And then once we've combined across areas, these are our sort of hard segmentation of where is each area located. And so this is a typical subject here. So we have 55B in between. We have the frontal eye fields above it and the premotor eye field below it. So 55B has got light myelination in it, and the two areas on either side have heavier myelination. 55B is a part of the language network and not a part of the dorsal stream network, whereas the eye fields are a part of the dorsal stream network. So these are the type of features that the classifier is being given to use. And here's an example where we have a different topology in a particular subject. So 55B is actually shifted up here. So normally it's supposed to be down there, but you can see it's up here. Now we've got heavier myelination in the location that group average 55B is normally. And the dorsal stream network is sort of in that place too. And so it's good to see the classifiers identifying 55B in the correct location for the individual, even though it's not the same location that it is in the group. So we can see it here. Basically, it seems like the frontal eye fields and 55B have sort of swapped positions in this particular subject. It can also happen that this particular area can be split in half. So here again we have heavy myelin in 55B, but sort of spots on either side. Basically the frontal eye fields and the premotor eye fields have sort of joined together, and 55B has been split. So in 80 or 90% of the subjects, we see the reverse pattern. We see 55B as a single thing and the eye fields as being split. But in this particular subject, about 6% of the subjects, we've got it going the other way. So here's our original group parcellation here, and here's an individual subject parcellation. Once we've combined across the classifiers, there's one subject, there's another subject, and another subject. Here are some probabilistic maps that we can make from the individual subject parcellation. So some of the areas like V1, M1, 3A, 3B, we have very little variability across subjects. Others we have more, some of these higher areas and more higher cognitive regions. And we can very nicely reproduce these probabilistic maps. So the top row is the 210P data set, and the bottom row is the 210V data set. So once we've trained the classifier, we can just simply apply it to generate individual subject parcellations. So once we've trained the classifier, we've generated a couple of weighting matrices that apply for each cortical area that allow you to distinguish that area's multimodal fingerprint from basically all of the fingerprints that surround it. So once you have that information, you can simply apply the classifier to find the area across subjects. And here once we've generated those individual subject parcellations, we've averaged across subjects to produce probabilistic areas. We can reproduce the multimodal parcellation at the group level very well as well. So these are group maximum probability maps. You heard about those from Simon already. For the 210P and 210V groups. Here we're displaying the areal boundary gray ordinates only. So if you see something that's blue, that means it was a boundary in 210P. If you see something that is red, it was a boundary in 210V. And purple is basically both. And you can see it's mostly purple. So the two groups show very similar parcellations, despite the fact that one of those groups was not used to define the parcellation at all or to train the classifier. So these boundaries are in very high agreement. Actually, the correlation of the parcellations is 0.97 at the group level. And this is in line with what we see for the dense map, group average dense maps for the various modalities is very high in HCP data if we do the careful processing. What about how frequently can we find each area in each individual? So we're going to call a hit or detecting an area if its size is within 1/3 to 3 times the group area size, even if it's misaligned with the group area location. And the detection rates in 210P are obviously very close to 1, which means we find each area and each subject almost all the time. There are a few places where it's a little bit below 1. And the same is true of 210V. So we're able to find the individual subject areas in these new group of subjects that we didn't use for parcellation or training. And the final validation is are these areas actually statistically significantly different across their boundaries? So we're going to do paired two-tailed t-test between area pairs for each feature across subjects. And we're going to use pretty stringent thresholds here. So we're going to have an effect size threshold. Basically, Cohen's d is a measure of standardized effect size, and greater than 1 is a really big effect. And then we'll also use a p-value of Bonferroni correction of this number of comparisons for a two-tailed test. And we're going to combine across feature categories. So we've got our myelin information, our thickness information, our resting state information, and our task fMRI information. So all of these modalities should be independent. So most neighboring area pairs have very large and significant differences across more than one category or modality. So we have plenty who are different across all 4, quite a few who are different across all 3, and not really that many that are only different across one modality. So what do you need to know about the HCP's multimodal parcellation and individual subject areal classification? We're going to release this parcellation in the future after we've published it, which we're working on right now. Additionally, we're going to release parcellated analyses, parcellated connectomes, task activity matrices, et cetera, for each subject. The software for computing gradients is already available in Connectome Workbench command line tools. You can visualize multiple modalities and parcellated data sets, SIFTY data sets very easily with Connectome Workbench. And the code to apply the areal classifier to new subjects, your own data, and the training data will be released in the future. And again, the images you'll need to acquire to make use of this process are these same T1, T2 fMRI and field map. And we have released already some other approaches to parcellation by the HCP focused on ICA. And I'll take a few questions, but I'm a little over time.
We have time for one question.
That was a great talk. So did you compare your probabilistic parcellation maps to the probabilistic map from the histological slices, which we saw in the earlier talk?
So we saw a little bit of that earlier in my talk, where we're comparing the Myelin maps and the motor and somatosensory maps. So yes, when they're available and when they're available on the surface, where they've been taken from individual subjects mapped onto the surface and aligned across the surface we have, and they're actually very nice agreement there. When they aren't available on the surface, unfortunately, the precision of the boundaries that you can see in the volume relative to the surface is not very good. So there you're a little bit more limited in what you can do. But for those areas that have been mapped onto the surface, the agreement is actually quite good with the cytoarchitecture.
Thank you.
All right, we're a little bit late, so if you have other questions, you can ask Matt directly during the break.